I'm such a goofball. How are you holding up, Ankita?
Pretty good.
Yeah.
I'm tired.
Yeah, I imagine. Well, we're just gonna sail through the day, like we're sitting back on a floaty thing on a river in the sun. I promise. There will be very little harassment. Well, I can't promise that. There might be some harassment. There could be harassment. Hello, Sadana. How are you?
I'm good. How are you?
Good. Can you do this? Whoa! This girl's Vulcan. She's definitely Vulcan. Lehan, can you do it? Oh, Astra's Vulcan too. And Keith, oh, you guys are all, oh my God. That's how you got into Cosmos. You guys are Vulcans. Live long and prosper, right? Unless you do it with the left hand, in which it means the opposite of prosper, which is go broke, and the go broke and die young. Don't do it with the left hand. Always right-handed. Celine, you're looking good today, different. Rohan, thank you for your wonderful background. Excelente. Let me see here. Got to be able to see it. Hey, Lucas.
Hello.
You do this? You guys are all Vulcans. Max?
I can't do that.
You're probably a different species. Probably a different species. Okay. What is a Vulcan? Means you're from the planet Vulcan. Means you're like Spock. Who can give Astra a better definition of Vulcan? There must be some Trekkies here. Isn't that on the Cosmos application? You have to be a Trekkie. Alrighty. Let me see here. How many do we have? 5, 10, 15, 20. Oops. 5, 10, 15, 20, 23. Yeah, pretty full room here. It's better than my usual, chemistry course, you know, because when you guys get into college, you're on your own, man. You go to class, nobody cares, right? Nobody keeps track. So a lot of us choose to skip class and just, be on our own recognizance or whatever. But, it doesn't, I feel like you're doing yourself a little bit of a disservice, but it doesn't really matter that much. But I can say for myself that there were a certain number of profs who really just blew me away. throughout my career, and it was equal numbers at Santa Cruz, where I did my bachelor's, and then I got a master's at Fresno State. Had some amazing professors there, just amazing profs. And I got my PhD at North Carolina, and just amazing profs there too. Although at North Carolina, they were much less focused on teaching. They were just like, come and do their job, and boom, they're out of there. Like they leave at the speed of light. They're like, like you're not going to catch them. But in any case, good to go to class. So thank you for coming to class. So I promised. Hey, Emma, can you do this? Another Vulcan. Gotta get this stupid camera out of the way somehow. I can't do it. Maybe I can. I'll do it later. I have to lean over to see Celine. Yeah, but on the other hand, when I'm talking to Celine, she'll get the impression that I'm talking right to her because she's right behind my camera. So I promised last time to talk about surface plasmon resonance. And I'll use this as a sort of a takeoff point here. I could actually even, oh wait, I gotta share my screen. Whoops, gotta share your screen, Roger. Share your screen. Screen one. Right, there we go. I'll present this. There we go. Hold on, I'm arranging you all here. There we go. So I'll talk to you today about surface plasmon, and we'll have let's put it different. Let's have a conversation about surface plasmons and nanoparticles in the context of, again, DNA hybridization and that type of detection, right? So DNA hybridization is pretty wild, right? Can you imagine, like, it's like a string of beads or whatever, right? And they're very long, and they're kind of flowy, but when they come into diffusional contact with other strands of a similar or of a complementary composition, they just zip right up. And it just amazes me how fast that is. Just because individually, the things holding the duplex together are hydrogen bonds, right? So hydrogen bonds are, they're fairly weak. They're not like covalent bonds, right? So, it's kind of like the suction cup of the molecular world, right? You can weld things together, and that's a good covalent bond. But if you just suction cup something on, that's a hydrogen bond, right? It's reversible. And DNA duplication is just all these hydrogen bonds, just one after the other, right? And we saw in the prior slide, for example, that there's a difference between, this is, how many base pairs is this? Oh, come on. I can't remember how many base pairs this is, but this is a single base pair mismatch creates, in like an 18-mer or something. It's got to be at least an 18-mer. And then with a single base pair mismatch, you can go from 90% hybridized to 10% hybridized at a given temperature. So, just as a little aside, that condition is referred to as stringency. So highly stringent conditions are ones where proper base pairing will be favored and mismatches will be disfavored. So like 40 degrees here, you know, body temperature is 38 here, right? And there's also salinity to consider, and then other stuff in solution that can be hydrogen bonding. But all of these components have to do with the stringency of the base pairing. I also there's some simulations in that you can see on YouTube, and there's a few groups that do this type of thing, and they do molecular dynamics, right? Which is it's like low quality simulation of molecular events, right? Molecular dynamics don't solve the Schrodinger equation every time. They solve it in certain cases, and they mash the pieces together, and then they kind of then they compute the properties, and they get the electrostatics and the mechanics and what, right? They can calculate the bond rotations. And there's a bunch of good movies where they can put these together, and they can display these very cool biochemical processes. And I'm going to show them to you right now. Let's see here. I've got to get a tab. Oops, come on.
Are we supposed to be seeing something else other than the title screen for the surface plasma and resonance?
Oh, hold on. I'm going to get that for you.
Okay.
Oh, it's still on the PowerPoint, right? Sorry.
Yeah, because I was just wondering. It's fine.
Okay, hold on. Let me get it. There we go. Can you see?
We can see your browser right now.
Okay, good. Let's see here. Now you're gonna see my. Let's see. Let's see here. I have to find. My library, and then I have like playlists or something, right? And I made a playlist for y'all called Cosmos 2021. Maybe it's watch later. No, it's not a subscription because I made it up. It might be in the library here somewhere, right? Playlists. I don't know why I don't have playlists. That's not right. Uh-oh. Oh no, this is the wrong person again. Oh, this is the wrong person. Erg. Switch account. There. No, this is not right. This is going to be stuff that I created, right? Yeah. Erg. Library. Ah, here we go. Here we go. Here we go. Cosmos 2021. Oh no, this is our stuff. Sorry. Oh, it's probably in my top videos here. Yeah, here's like, have you ever seen an atom? We saw that one, right?
We have seen this one, yes.
Here's a good one. Here's a good one. The electron transport chain. Let's see if how this one looks. Let's see. Can you guys hear it?
I don't think so.
I can hear it now.
Be able to hear it now.
Yeah, I can hear it now.
Yeah, it sounds good.
One of the
Yeah,
key
yeah.
roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix.
So this is what
The
you would
membrane
describe as a
also
very coarse
contains a
grain
large protein complex
simulation.
called F1F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die.
It's dying.
Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. proton pumping requires energy, and the four protein complexes get this energy by transferring electrons through a series of coupled reactions.
Electron transfer, yeah.
This linked process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to
And the
the
electron
bottom.
disappears.
First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons.
Bioenergetics, baby.
The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in two important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons.
So,
Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water.
yeah, that's
The
excellent.
proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain.
My electrons,
In
oxygen.
this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
So cool. So that's a coarse grain simulation. Now, how about check these guys out. This is WEHI. I love the sound effects in these guys. These are slightly more finely grained simulations, right? And this is some sort of ATP, I'm sorry, DNase. It really is that loud. Yeah, can you imagine having 1.8 meters of string, having to pack it all into the nucleus of a cell? And then, oh, I need nanometers. $500,673,000 to 500,674,382. So weird. It's creepy. It's also squishy. Oh, here's another one. This is awesome. They don't even show the ingredients. You know, they don't even show the amino acids going in to get incorporated, but they have to be, you know? Vitamin D is just a small molecule in there, you know? Transcription, oh my. Now you could transcribe duplex DNA. Duplex DNA to single-stranded RNA. Isn't this shit weird? Kind of like looking to the interior where the base pairs form, you know? Your genome contains 3 billion base pairs. Yeah, how do you know? I bet you yours contains only 2. So the epigenome or the epigenetics are methylation, phosphorylation, and other modification of DNA that they're actually, some of the information is actually passed on, and it activates certain genes and suppresses other genes. How do we know all this, you know? It's fairly speculative, you know? Fairly speculative. You know, parts of it are known very well. A lot of structures are known very well. A lot of it's molecular dynamics, simulations. Okay, now. Stop. Okay. Stop. Okay. Gotta get back to the boring stuff. We were talking about using DNA hybridization to find DNA, to find other DNA molecules, right? So we're using one of these DNA strands, hoping that it just wraps up with another strand in solution. And the way that is done generally is to, can you guys see my presentation? The colloidal gold enhanced surface plasmon.
Yeah, we can see it.
Excellent. Thank you. Good. Oh, you know what? Anyway, I think my headset's not connected. Bugger. Okay. Don't need these. Not helping. Okay. So so you know, you use chemistry and you modify the end of a DNA. You know, you basically you make a DNA strand. You know, you use all these organic chemistry tricks. You take beads and you build up little strands of DNA, one after the other. You use beads because you can immobilize the beads and you can rinse them. You can do a reaction, then rinse everything off, then do another one, rinse it all off. That's called solid phase synthesis. You don't get a whole ton yield, but you can do these purifications really quick, right? And so you can make or buy DNA oligomers, you know. In this case, they use a 20-former, right? That means there's 24 base pairs. And they attach a 20-former to a nanoparticle. Then they attach the complement to that strand to a gold surface, right? And they do this all in solution. And they have about 10 to the 9 molecules per square centimeter. So 10 to the 9 is about 10 to the minus 15 moles per square centimeter. So that's a very, very, very low coverage. But still they work, right? And so here's a picture of the way that it works, right? You've got you can either do do a sandwich assay, right? Where you have probe 1 on the surface, probe 3 on a nanoparticle, and then target 2 in solution, right? And then you can either do it what they describe as amplified or unamplified detection. And I'll try to explain the detection in a moment as well, right? And what you see here is a surface plasmon resonance curve, right? And one way to do this is to look at the reflectance of a surface as a function of the angle of incidence of a light beam, monochromatic light beam. Typically, this would be might be 632.8, which is a helium neon laser, or 750 nanometers or something like that. And the light reflection has this interesting quality here. It bends up, bloop, and then this region up here is called total internal reflection, right? And then it dips down, and then comes back up. And this dipping down is a very unusual phenomenon that's due to something called surface plasmon resonance. Okie dokie. So I'm going to talk about surface plasmon resonance now. And going to use a different presentation that I have. That. Can you guys see the surface plasmon resonance overview?
We can.
Yes, sir.
Okay, great. So in surface plasmon, you're taking advantage of the collective oscillations of the free electrons in a metal. And that's it's kind of a funny thing to say, right? What are the collective oscillations of the electrons in a metal, right? So remember when we were talking about electrons and how they can flow through a metal, right? That there's the Pauli exclusion principle that prevents one electron from moving unless there's a vacancy around it, right? Well, that perspective is a little bit out the window here, right? And what we're thinking about now is we're sort of zooming out and we're thinking, okay, so there's a whole bunch of electrons and they're bound in this metal and they can all flow around and they all exert an influence on each other, right? And you have to think about the balance of their energy. Like they're attracted to the nuclei of the metal, but they're repelled by each other. And so in this, sometimes it's referred to as a gas, or sometimes as a liquid, right? Because if you disturb the electron density in one place, it'll go up and down, and it'll make waves, right? Now, when light reflects off a metal, that is you could describe that as an elastic interaction, where the photons come in and they leave with all of their energy, right? And that type of interaction does not excite collective oscillations in electrons. But you can tune the wavelength and the frequency of light such that it does excite collective electrons. And that's what I'm going to try to describe to you now. Now, I don't know how, but oh my God. I think I have a mistake here because Fresnel lived between 1788 and 1827. And then I have 1891 for his equations. So I have a little mistake there. So this is Fresnel, I guess he was a contemporary of Maxwell, maybe a little before Maxwell and Faraday. But he and Huygens and others, they did amazing work to calculate the reflection of light. You know, they knew about this thing called refractive index, and they could measure the angles of light, and they knew about polarization. So light polarization, there's for light reflection on a surface, there's an S and a P polarization. All right. So these are the equations for the real part of the reflectivity. And the real part is the lossless part. This is for specular reflection, like a mirror-like reflection. If you have loss, there's an imaginary component here. And you need loss to get a surface plasmon. Okay, so what is a surface plasmon? How do you excite it? How does that look? Well, most commonly what you use is called the Kretschmann or the ATR configuration. The ATR stands for attenuated total reflection. And in ATR, you've got a, let me see my cursor. So,
Wait, Dr. Terrell?
yes.
So I kind of missed it. Can you please explain what surface plasmon resonance is again?
Yeah. So I'm sorry, I'm sort of creeping up on it here, but surface plasmon resonance is when the light from a thin metal film is absorbed. The light reflected off a thin metal film under conditions of total internal reflection is absorbed.
Absorbed by like an atom, or?
It's absorbed collectively by the electron density in a metal film. Or similarly, you could say, and this is termed localized surface plasmon resonance, it's light absorption by the collective electron density oscillations in metal particles. Right, so the metal particles that you make in lab, the gold, have this beautiful red, this wine red color, and it's completely different from the color of gold in its bulk form. You know, that's that sort of beautiful gold yellow color. But when you divide the gold down into these tiny units, their color changes. And the color changes because the particles begin to interact with the electric field of light in a very special way, in a resonant way. And what I'm trying to describe here is how that resonance is set up in a thin metal film. Okay, so let me just describe this case that we have here. Right, we have a prism. This is a three-sided prism, and on one of the faces of that prism, oops, there we go. Come back. Pointer. Pointer options. Laser pointer. There we go. You have light coming in, and it's coupled into the prism, and then it bounces off of a thin metal film here, and then it leaves, right? And you observe the light that comes out. And basically you measure the intensity. Then what you do is you change the angle of incidence here, and you look at the angle dependence of the reflectivity. And before the light comes in, you use a polarizer to make sure that the polarization on the input is all out of the plane of incidence of the metal. So this is referred to as P polarization, right? P for perpendicular, although I'm not really sure what that means, but that means that the rays that come in have their electric vector in this cross section of the surface, such that, you know, as the wave comes in, classically it's described as an extended wave in space, which can then project onto the surface of the metal, right? And then that projection actually creates a new wave in the metal plane that's expanded relative to the first wave, right? And let me show you how that works. Come on, you can go, come on. Next, there we go. Let me see if I can do this here. Oh no, you have to work. Play. Rats. No. This is supposed to play, you guys. Rats. Anyway, I'm so sorry. But basically, as you change the angle of incidence, the intersection of the crests with the metal surface accordions out. And so what happens is that at a certain angle of incidence, you hit a resonance with the free electrons, right? And so the magnetic component is in the plane of the surface. The electric field is in the Z direction here. And the light comes in, and then it excites these electric field lines. And the metal here is right at this interface, right? And you can see that on the prism side, you get these broad arching, they're referred to as evanescent waves. Then on the metal side, you get these shorter waveforms, right? But if the metal film is thin relative to this evanescent wave thickness, then you can create this wonderful sensor that you present to the world, which has this plasmonic evanescent field that projects into solution just a little bit. So let me, this isn't going to play either, I doubt. Oh, come on. Rats. This is my experiment and darn it, I was the first person to do this. Anyway, but what you can do is you can create an angle wavelength expansion of this whole surface plasmon thing, right? And the upshot is that okay, so I'll do this later basically, but that you can, it's just too complicated when I go through it here. So let me go back to let me go back to this description of things. So basically, if you look at the total internal reflection curve before and after the injection of these DNA molecules, you can see a tiny shift in the plasmon curve going from case A to case B here, right? And that tiny shift is because something has bound to the outside of the metal film, the inside of which is being irradiated with light. So here's a surface plasmon curve. The black one is the blank. The dashed line is with having injected the target DNA, and you can look just at a single angle. If you look at this angle at that point A right there, then you can see that the reflectivity increases by about 1% when this DNA binds to the surface, right? However, if that DNA is bound to a nanoparticle, then you get this enormous shift all the way from A to C. And this is the signal that you get for this single base pairing event, or this single, it's not a single base pairing, the single duplexation event that happens on the surface when you've got one of the partners bound to a nanoparticle. Does that make sense, more or less, kind of? I feel like I'm being super boring today because I'm trying to take you on a magical mystery tour, which is equivalent to an inner tube going down the Truckee River from Lake Tahoe in the sun in the summer, where there's cool water and sunshine, and we're just floating and bobbing along the waves and drifting down the river. But all the
Dr.
while,
Harris?
somehow we're learning. Yes?
So I was wondering with the,
Yes.
so what happens when the angle of light is changed?
So, you know, have you guys heard about total internal reflection?
I have heard of it, yes.
You've heard of it, right? And so it's like, if you have a beam of light in a, let's say that down here was glass and up here was air, let's say. And then I've got a beam of light. If the beam of light is coming straight up, then most of it goes through, right? But at a certain angle here, that'll be the glass air interface will behave like a perfect mirror. And it'll just reflect the light, all of the light, right? And at that's called a critical angle. And that critical angle for this interface is right here, about 49 degrees, right? And then beyond that critical angle, you can see there's this large attenuation down to maybe 5% total reflectivity, right?
Mm-hmm.
And it's this this dip in reflectivity that is the signature of the excitation of a surface plasmon. And I'm so sorry my animation didn't work. I don't know. It makes sense that it didn't work because I'm on the wrong computer today, so I'm sorry. But
Fine.
yeah, but in any case, this minimum point is a place where the angle is tuned such that the frequency of the light, which is fixed, and its wavelength projected onto the surface, which can accordion, matches the frequency and wavelength of the surface plasma in the metal, right? It's sort of like You know how if you drop a pebble into water, you set off these water waves? Well, there's a resonant frequency for those water waves, right? And it's the same is true of the electrons in a metal. It's just much higher frequency. And so here at 54 degrees, there's, you're probably at like, 60% of the speed of light if you take the frequency times the wavelength. And so you basically, you slow the waves way down in this high index prism, and then you speed them up by angle tuning, and boom, you can hit this resonance right here. And so that resonance corresponds to the matching of the electromagnetic frequency of light to the electromagnetic frequency of the surface plasmon. The plasmon is the plasma waves in the electron density of the metal.
Okay, and when you change the angle of light, it just changed the frequencies at which the surface plasmons are at?
You're on exactly the right track. That's a great question. And I would reword it as following. When you change the angle, you change the projected wavelength of the light in the surface. It's I could probably draw you a picture. Can probably draw your picture here.
And that would change the frequency of the surface plasmon?
In a limited way, yes. So the surface plasmon kind of has its own frequency, right? And actually the surface plasmon frequency is affected by binding events at the gold surface, right? Let me see here. Oh yeah. I'm going to try to share my little stupid document camera here. There we go. Cool. So let's say here's the metal surface, and here's the light wave coming in, right? And then the projections of the maxima in the light wave are actually onto the surface. They're actually this wavelength here is lambda. This is lambda prime, is lambda over sine of theta, where there's theta, right? And so the frequency of the plasmon is driven at this same frequency of the light, right? That's fixed by the light frequency, right? But the lambda prime is equal to lambda over sine theta. So you can adjust this. Oops. Right. So that's what you adjust experimentally. And then you look at it's when the frequency of the plasmon is right and the wavelength of the plasmon is right, then when these two are correct, then you get the resonance. And that's at the bottom of the reflectivity curve. Make sense? Cool. Excellent. So, oops. So back to this then. What I'd like to share with you all about it is that by using, by using this colloidal, by using the magic of biochemistry coupled to the magic of nanoparticles and surface plasmon resonance, you can detect these individual these duplexation events happening on a surface. And what these folks did was they made, this black square is a section of a gold surface. And the gold surface is imaged, and you can see these circles. The circles are places where that gold surface has been modified with the oligomer of choice here, right? So we've got 0, 1, 0.1, and 10. 0, 1, 100, and 1000 picomolar. Wait, no, 0, 1, 110,000 picomolar. And what the authors have done is they've expanded the laser beam such that it covers this square centimeter or so. And then they've just spotted the slide in these regions, and they're imaging it at a constant angle. And they've set up the angle such that the difference between where the bound and the unbound target is a large reflectance difference, right? So they're at 53.5 degrees, and then, then they're imaging at 53.5 degrees, such that when a binding event occurs, it makes a bright spot. It's called SPR imaging. And then you can actually see the places on the surface where these oligonucleotides have bound. And then you can get a sort of a calibration curve here on this is on a log scale. 10 to the minus 12, 10, and 8. This is 10 nanomolar here, and this is 1 picomolar. So this is a very wide range of concentrations. For some reason, they don't use these data here, I guess. Probably they fell off the line. Anyways, but anyhow, I think this is really all I wanted to say about it. Was that it's a very interesting confluence of phenomena from surface plasmon to nanoparticle, and to the coupling of these two types of behaviors into a very amplified response. And again, you can, what's being done now, there's a couple of companies called Affymax and Affymetrix, and that stands for, Affy is for affinity. And these two companies are developing this type of sensor for determination of a whole variety of proteins and nucleotides and other components of plasma, blood plasma. And so you can, the idea is that you can take a chip like this, and you could spot it in all these different ways, and then get a blank, and then flow this solution, this blood solution, or this plasma solution, or this whatever solution you've got over it, right? And then the places on the surface where there's a binding affinity for those targets, they'll light up, right? Because that'll change the plasma resonance condition, and you'll be able to see that move from resonance to off resonance, and then you'll be able to quantify the amount of analyte in a given on a given gene shift. And that, my friends, is one of the only ways to do it, right? To know when things are binding in a biochemical setting is you've got to have, generally you've got to have some way to transduce that binding signal out into the big old real world that we live in, right? And this is one way to do it. There's not an unlimited way, unlimited number of ways to do it. So I think I've probably bored the pants off of you today, but how are you doing? Yeah, kind of like, was it like,
It's just it is just a lot. Like it's just very, very, very much information.
Yeah, I'm afraid it was a little bit too much information. Yeah, I've got to break these down into some better little sort of teaching points. I mean, I'm afraid that, I'm basically obsolete now, because I mean, except for the particular collection of information that I have, I mean, YouTube is just replete with great videos on different subjects like these. And like, oh my gosh, it's just a pleasure to be able to browse through like all these different YouTube sites and listen to experts talk about stuff. It's amazing. And it's all free. Tell your parents. No, I'm just kidding. Don't tell them. Don't tell your parents. So are there any questions maybe, or?
I meant to ask this earlier, but I didn't want to interrupt.
Oh, sure.
Do you think you can go over a little bit more what collective oscillation meant?
What plasma oscillation is?
A collective oscillation.
Oh, collective oscillation, yeah. So, so when you talk about electrons, right, you can certainly have a single electron that's oscillating, right? And it just means it's going back and forth. That's all it means, right? And, but when you have a collective oscillation, what that means is that you don't even think about the individual electrons. You think about their behavior as a fluid, right? Now, this is the one thing that we kind of have to get into our minds about metals, is that the electrons collectively behave like a fluid. Because every electron is attracted to every nucleus in the metal, right? And the electrons are largely free to move around the metal. Not in every direction, but there's have a great deal of freedom to move in the metal. So if you, for example, if you take a metal and you fire an electron into it, you have like a little electron gun and you fire an electron into it, you can observe plasma oscillations. You can observe them in the metal. Like, I don't know exactly how they're detected. I think it has to do with the emission of other electrons. I don't, it's not quite the same as the quantum corral that I showed you, right? Those are quantum states, right? Those are very, very small. But the collective oscillations are much larger, and they have more like the wavelength of light of hundreds of nanometers, you know?
And is that what you said acts like a wave or a ripple in water? Like when you
Right.
shoot a single electron or
Yes, exactly. Yeah, they behave like water waves.
cool. Thank you.
You're very welcome. Thanks for the question. Sometimes, you know, if you go to scientific talks and stuff, a lot of times the talks are a little bit dry, maybe a little bit boring, you know, not always, but sometimes. And I always really try to think up good questions during the presentations so that I have something to ask at the end, because it's really a kindness to the speaker, you know, because like we you know, when you're speaking, you're trying to do your best to convey a topic, and a lot of times you just fall flat on your face, you know. And if no one has any questions, it's almost like saying, I didn't even get enough information to ask a stupid question, you big jerk. You just wasted an hour of my life. So thank you, Astra. Thank you, Vigo. Thank you, everybody, because I know everybody's paying attention, you know, as much as you can. So I really, really appreciate it. But special thanks to Astra right now. So more questions perhaps, or?
I was also wondering, in
Yeah.
SPR, when light exits the prism, is there any use to its detection, or like, I guess, what's the application of the light that enters and exits the prism?
Oh, yeah. So, you know, the light that enters and exits. If you, you can see this, can you see my mouse cursor on the screen now? Maybe?
Yeah.
Yeah. So these spots that you see, they will actually show up on a screen, right? And this is just a representation of the reflection of the light off the surface. So, you know, it's probably like orange light or red light or something that's just shining on a screen, right? And then there's the detectors on the screen, right? Or focused on the screen or something. And so you can see the light that's being reflected, and you can detect these photons very well, you know. And there's always these weird defects, like this circle blue blobble thing. I don't know what that is, but they're always there, you know. And so the light that comes in and out is just like how you can see the resonance, right? As the light angle goes up, then, oh, wow, it's gone. And then it keeps going, oh, no, it's back. And you go back and if you look at different angles, the light disappears, you know? It's kind of like a freaky mirror that way. So like at a certain angle, it goes dark, but then at another angle, you can see it. All right, guys. So thank you very, very much for your attention. And I look forward to seeing everybody very soon, tomorrow. And are you guys have like a little thing that you're doing some independent research on, right?
Yeah, it's for our final presentations.
Mm-hmm. Okay, so what's today, Wednesday? So we've got, if any of you want to spend any time just chewing my ear about that, let me know. And I can set up some office hours, or we can do it during our regular period, or whatever you like. Okay. Awesome. Okay, good. I look forward to hearing from you guys. I will see everybody tomorrow.
Thank you so much.
Thank you.
Thank you very much.
Thank you.
Have an amazing day.
Awesome. Thank you.